welcome to NRGene's webinar series. My name is Gal Ramot from the marketing team here at NRGene, and I'll be hosting this session. Today we'll be hearing from 4 leading researchers about the advantages of pan-genome and the drawbacks of working with a single reference genome. It's my pleasure today to present the speakers. With us today is Professor David Francis from Ohio State University. His program releases tomato varieties and parents for the processing tomato industry, and his group has been instrumental in developing SNP resources for the plant breeding applications. Also with us today is Chris Dardick from the USDA ARS, who is a lead scientist for the Genetic Improvement of Fruit Crops Unit at the Appalachian Fruit Research Station in Kernersville, West Virginia. Also joining us is Dr. Paul Matthews, who has over 20 years of experience as a senior researcher at Hopsteiner, where he has developed molecular breeding in hops, founded the Humulus Conservation Project, and the Pan-Can Comparative Genomics Group. Paul is also Chief Science Officer at the Analytical 360, Washington's premier cannabis testing lab. Last but not least, our very own Dr. Paul Comment will be with us today. He is NRGene Senior Technical Specialist. Paul has extensive knowledge in molecular breeding and biotechnology with over 25 years of industry experience. As an introduction to today's webinar, I'd like to introduce you to the topic of utilizing a genome reference in research and breeding. Since the early 2000s, when genomes were starting to be assembled, it was recognized that one genome did not represent all the genes found in a given species. This was also recognized early on in plants and is well characterized in the example of maize. Studies show that many regions of the genome are not orthologous, and this includes genic regions. The need to sequence and assemble multiple genomes to represent the genic content was first recognized in bacterial species, and the use of the term pan-genome was coined to represent the collection of all genes found to exist within members of a group or a species. In this graph you see here, we can show in maize that as we de novo assemble and compare diverse varieties of maize listed on the x-axis, we continue to identify unique genes counted on the y-axis and as identified by the red line, termed variable genomes. The blue line represents the shared genic space, termed the core. This added variation also applies to the intragenic regions as well. The understanding of the pan-genome has significant ramifications on gene discovery, genetic mapping, genetic marker development, genome modification such as gene editing, and breeding strategies. Next up, we will hear from Chris Dardick on his perspective and research utilizing genomic information. Chris, the stage is yours.
Thank you, Paul. Just to give you a little bit of background, I've been working on fruit crops, mostly fruit trees, for the past 15 years. And we run both breeding programs and functional genomics and biotechnology as part of that. So to give you a little bit of background on peach, let's see if I can advance the slides. Peach has been a model species for the Rosaceae family and for fruit crops for some time for a few reasons. One is it's a very small genome. It's diploid, and there's excellent genetic resources for it, genetic maps. And the genome for peach was first sequenced about 10 years ago, and it was done from a double haploid, so it's a very high-quality genome. And from that, it became kind of, I think, the sort of reference genome, not only for peach, but also for other prunus species, which we've been using, and I'll show you a little bit of that as well. So over the past decade, and this is paralleled in many different crops at this point, a number of resources and tools have been developed using that genome in order to enable marker-assisted breeding. And one of those has been things like SNP chips and other types of marker platforms so that you can better select from your breeding populations. that information has also led to not only marking specific traits and genes, but also potentially the use of for functional genomics and identifying actually causative alleles. And that's where most of our work has really focused. Let's see here. Apologies. There we go. So around the same time, shortly after the Peach genome became available, we began using a different approach for mapping, and that's a bulk segregant type analysis. So this was based on, again, the reference genome. And we did this not using any predetermined SNP markers, but by sequencing pools of individuals based on trait, and then using that information to find alleles that were specifically associated with a certain trait, so that their frequencies, for example, would be higher in pools of one trait versus another. And so that information we can then use to develop a map and a graph and identify the location. So it doesn't require predetermined SNPs. We determine them based on the reference genome assembly. So here I have some examples of 4 different maps that we've developed for different traits over the years. And the way these are generated is we have variant frequency on the y-axis by chromosome position. And so you'll see clusters of sort of associated SNPs based on their frequencies. And when you they indicate a particular map position based on a group of SNPs and where they cluster. So this has worked very well, but we have run into, in a number of cases, some difficulties, and that has to do actually with the actual reference genome. And in the next slide, can show you a particular example of where we had, just to compare the map location that was generated from a more of a SNP panel-based approach versus this bulk segregant strategy, which is now commonly referred to as QTL-seq. I apologize in the figure here. Actually, the labels here are off. This sort of vertical line here, you see a cluster of SNPs, which are indicated with the diamonds. That's actually where the original map had positioned it. We positioned it to the right at the peak of this curve using the bulk segregant approach. And what we later discovered is the primary reason for that was just simply the very small number of SNPs that were present near that locus. So these sort of hot spots or regions of SNP density were kind of distorting the results to some extent. Here, and on the next slide,
Chris, Scott here. How about I move the slides for you?
Sure, that would be fine.
All right.
So if you can jump to the next slide. Okay. So the other challenge we had from using the reference genome was when we were trying to identify what the causative alleles are. We in some cases found that they simply weren't represented in the reference genome in a way that made them easy to detect. So this is an example of one of the genes we mapped, we call TAC1, or it's from a pillar type architectural trait in the tree. And because of the way that the reads assembled to the reference genome, we ended up with actually there was an insertion within the turned out to be the causative gene, which wasn't detected by any of the indel or SNP finders or anything we were using. And that had to do with just simply the way the reads had assembled around that particular insertion event. And in fact, to this day, we still don't know what that insertion is because we've been unable to use PCR or any other molecular approaches to actually find it. So that's just an example of, I think, where missing information from the reference genome was, in this case, particularly an issue for us. Next slide. So in this figure, which looks a little bit complicated here, I'll try to go through this. But this shows, I think, a little more clearly how varying SNP densities can distort your mapping data. So again, on the top here, the graph, you see the blue dots. That's actually another QTL-seq map we got for a particular trait. And in this case, it was actually a QTL, so there were two loci, one around 24 megabases and one around 9 megabases. And below that, what I'm showing are in sort of that pinkish orange, those are individual SNPs, unfiltered. So these are the SNPs we identified relative to the reference genome across the entire chromosome. And then you'll see in blue are the SNPs that actually show linkage or association with the trait we were interested in. So if you look to the right, you'll see a nice, where we had our strongest peak, you can see this cluster of associated SNPs. And I have a black dotted line drawn across the center. It actually should be shifted down a little bit at 50%, but that's essentially the random association line. So anything at some point, it becomes significant, close to 70%. But you can see on the bottom how essentially the frequencies of all the SNPs in a truly linked site will shift upward. And that has to, and the variation you see has to do with the sort of random sampling or of lots of different SNPs. So their associations will depend on some random events in terms of the coverage and the way the SNPs are called. And you can see on the left-hand side where we have what look like associations, and it's not as clear as to whether they're truly associated or not, because all of our SNPs show up where we have very high SNP densities, and we have a lack of SNPs where we have low SNP densities. So that general feature can result in distortion of your mapping. The other thing I can say about that is the varying SNP densities, at least from the experiments we've done, are not necessarily consistent. They depend a lot on the reference genome, because that's what you're essentially identifying your SNPs from. So these aren't necessarily hot spots that are conserved across speech. They change from one source of germplasm to another. So this is a case where I think having a single reference genome, again, just kind of highlights how that can be, I think, a little bit misleading, or at least a limitation to trying to do this kind of work. So moving forward, we're now looking at doing similar kinds of work in other fruit crops. So one of these is citrus, and we have a new project to map HLB resistance. So Huanglongbing resistance, or citrus greening disease, is obviously a huge problem. And we have new germplasm, which appears to have real resistance to that disease. And this work being done by UC Riverside. And we're working with them now to try to do something very similar and map these disease resistance traits, which come from Australian limes. So the citrus germplasm is quite diverse. And this is a case where we're immediately going to a pan-genome approach, because we need multiple citrus genomes in order to ensure that we don't have this problem when we try to identify these loci, that we simply have missing regions or mapping distortion that leads us in the wrong direction, because this is a case where obviously it's urgent to try to find a solution as quickly as possible. So next slide. Another trait we've been interested in for some time, and I just kind of put this up because it's kind of fun, stoneless trait in plums. So the idea of developing like pitless plums or pitless cherries was something we've had for some time, and we actually have a naturally occurring mutant. So we had begun trying to map using the peach genome as a reference for plum. They're somewhat closely related, but this turned out to be kind of a disaster. This didn't really work for us at all. If I give the next slide. The reason is that even though the peach genome is a reference, we can assemble our reads to it, and we can find SNPs, and many of those SNPs are real. The complexity of the plum genome was just too much, and we couldn't really get any meaningful mapping data from that. So this is just an example of some of the fruit crops have these kinds of more complicated histories, and in this case, the plum we're working with, European plum, turns out has probably 2, maybe 3 progenitor parents, and it's a sort of more of an interspecific type of a species. So it is a hexaploid and has a very complex genome, which we've just recently sequenced and assembled. And even from that data, we found that it will be necessary for us to sequence the progenitor genomes as well to really understand the genome, the alleles, and how they segregate, because we have a lot of segregation distortion in this genome. So these are just a few examples that I wanted to kind of highlight as to from our own work where using a single reference genome has been limiting. Have the next slide. And just a couple of summary statements here. One, what we found is that a single reference genome only captures a fraction of the genetic content within a species or population, which we know, this is not anything new. It's just a question of how limiting that is to your work. And in our case, we're now beginning to see where that's becoming a real limiting factor. And obviously that missing data is an impediment to molecular breeding and functional genomics. And then in cases where we have broader genomes or crop species that have much more diversity, the kind of pan-genomic information, I think, is really critical to being able to do this kind of work. So with that, I will stop and introduce the next speaker. Can I have the next slide? So I'll turn it over to Paul Matthews.
Hi, this is Paul. Thanks for inviting me. I'd like to, and I may need help also with slide advance. So next slide, please. I'd like to introduce you to Humulus lupulus, which I think you all know is the flavor of beer, and you know it that way. But as a specialty crop, it has new and limited genomic resources. It's another problem plant, and I'll make many of the points that the other speakers will make about these niche species. Hop is dioecious. It has an XY system. It's self-incompatible, so there's no inbreds. So it's necessarily heterozygous. It has a large genome, about the size of the maize genome. And I'm going to show you that we've discovered a prevalent meiotic phenomena, translocation heterozygosity, which may lead to permanent large blocks of permanent translocation heterozygosity. There are polyploid breeding programs in hops, but it has a limited history of domestication, meaning small inbreeding programs, small effective population size, and we're introgressing wild subspecies. So we have the interest and are expecting large genomic diversity that will require pangenomics. One slide, please. Maybe I have it. Hops is the other member of the famous Cannabiaceae family. So actually, hemp, marijuana, is a much simpler genome and may end up being a good model. So we're engaged in comparative genomics across the Cannabiaceae, where hops and cannabis are the 2 economic species. There's 3 species of hops, and their Humulus lupulus is the one that's used for beer. It's the bitter products are made in trichomonas glands, as are the cannabinoids. Very similar systems, trichomes that exude what is now pretty well understood biochemical synthetic products called bitter acids in hops and cannabinoids, very similar biochemistry. There's 5 subspecies of hops that we're interested in having collecting for integrations. And I think ahead, please. Phenotyping can be fun in hops. We're interested in aroma and flavor, and hence we do a lot of deep chemistry, metabolomics, and that kind of phenotyping. But of course, we also do field breeding and cone shape and yield components, and plant architecture are also things that are target traits. But I'll show you actually today breeding for powdery mildew resistance. We all breed for disease resistance most of the time. We've created a number of genomes over the last few years. Early genomes were quite lacking in coverage. If you look at the progression in number of scaffolds, you can see we now have a very recent, in fact, just now published at HopBase and by our archive, the Cascade genome. Cascade's a variety of hops that's the most popular in the US. And we have about 2,300 scaffolds now after using Hi-C to put that together. Still, this is the genome is not fully covered. It's about 3.4, and the Cascade genome is not haplotype resolved. So it's been collapsed to a diploid, and we don't have the male and female components of the Cascade genome in hops, which is a diploid. Next, please. We've developed GBS SNP systems in Edward Buckler's lab, and we suffer with our large genome. We suffer with the usual problems with GBS SNPs, but additionally, because it's obligately a heterozygous species, we have uncontrolled parental genotypes. That is, both parents are heterozygous. We haven't resolved that. There are methods to do pseudophasing, which we apply. And we think because the founder effect, but also a phenomenon I show you, which is a non-homologous recombination during meiosis, we have very strong linkage to equilibrium and blocks of pretty meiotically inactive chromosome. And as I said, there's a number of genomes and the quality is improving. Next slide, please. So we have proceeded to do GWAS for simple dominant traits. For example, sex. You can see we have the Manhattan plots do have phase components, but again, this is pseudophasing. It's not really real. And we've, of course, got the suspicious candidate genes that everybody else has, and really don't trust this, and really not proceeding with analysis of candidate genes and looking at haplotypes across the germplasm until we have better genome, and we need haplotype resolution provided by pan-genomics. Next. And so we've also been successful in powdery mildew resistance genes. On the center panel, you see there's a number from different plants or various diverse sources, a number of characterized R genes, simple monogenic dominants in most cases. And we've created nested mapping families, which are, we often map in half sibs as families, which is another complication. Next slide, please. Yeah, thank you. And we've been successful in finding and then predicting the R1, R2, and R6 loci. But we don't really have allelic resolution, even though we have good pedigree. And we find that these are in different mapping families for R1, R2. We find they're on corresponding linkage groups. That is, it looks like these resistances from different sources are on the same chromosome. We really don't have the resolution to continue to seek candidate genes and follow this from pedigree. Next slide, please. And we also want new resistances for a number of diseases and have a rather far-flung conservation project that as we go wild hop hunting, and we're now integrating. We have screened for other disease resistances and additional powdery mildew resistances in exotic germplasm and are integrating that. So again, we're anxious to be able to look at the diversity across the whole breeding program. Next, please. And then I'd just like to comment on our last great complication when we started developing reference SNP marker systems by GBS. We noticed that the majority of markers are in very heavy segregation, a large degree of segregation distortion. 70-some% of markers are heavily distorted. And when we looked more carefully at clustering correlations between markers and finding linkage groups here by modulated modular clustering, and then projection of those correlations, we found that our linkage groups were not discrete and actually coalesced. Next slide, please. So, wrong way, yeah. So here are the 5 largest linkage groups, and when we project the correlations, we find that the linkage groups are sharing large sets of correlated markers. And then if we look at the nature of the markers in the sharing, we find here in blue and gray that it's the markers, the SNP markers that have very low minor allele frequencies or heavily distorted that are causing the sharing of groups. That is, it appears that these markers are on multiple chromosomes and shared. Next slide. And then we enlisted cytogenetics, who went back to the good old days and looked at cytogenetics with Hank Bass and Catherine Easterling. Next slide, please. And we found that the cytogenetics, this is a panel of various diverse hops. There's some wild hops here, domestics hops, and hybrids between the two. We found by looking at meiotic chromosomes in males and anthers, we found, and we can do this in 3D so we know it's real, we found a lot of non-homologous recombination. And here in this last panel is an extreme one where every chromosome is interacting at least with every other chromosome. And we find break fusion bridge cycles and segmental aneuploidy and many other hallmarks of sort of dysmeiotic phenomena. And we think these phenomena will lead to permanent translocation heterozygosity, which then becomes really unmappable. So the challenges, I'll just summarize. I've presented just problems with my favorite plant, some which may be resolved by comparison to other species within the family. So hop is a specialty crop. The economics are limited. It's necessarily heterozygous because of its reproductive habit. It has a small effective population size and breeding programs, but we expect large structural diversity. We do have a number of mildew resistancies and have proceeded to find alleles and are interested in wild integration to bring in more disease resistance to create horizontal resistance. But we're very concerned about our early results with SNP systems and effects on families, mapping families, recombinant families, that we also have this large scale and very prevalent translocation phenomenon. So I'll leave it at that and go on to Paul Chomet, who will, Dave Francis, who will present tomatoes. Thank you.
Thanks. I'm gonna start by thanking Gal and Yana, the NRGene team that's providing support for this webinar, and I'm going to thank the audience for bearing with us as we work through some of the issues with multiple organizers trying to advance slides. So if I'm not able to advance the slides myself, I'm going to go ahead and ask Gal to do so. So one of the things that I wanted to start out with immediately is acknowledge a recent paper out of a team from BTI and other universities that used a map to pan strategy and resequencing data for as many as 725 accessions. This is an important paper because it really highlights in tomato that there are a large number of genes, so close to 5,000 genes, that appear to be absent from the Heinz 1706 reference. So not only does this paper reveal non-reference genes, but it also suggests that there are variants that have been selected by humans during the domestication process. At the same time, this work is still relying on resequencing data and has not produced new reference quality sequence. And I'm going to try to present some data to you to convince you that having more reference quality sequences is actually going to be really important. And then another point that I would make is that even though there are 725 accessions, they're really focusing on domestication. And so there remains an underrepresentation of germplasm that's relevant to current improvement efforts, as might be defined by a plant breeder. So let's go ahead and advance to the next. There we go. Okay, so one of the things that I wanted to tell you about with this slide is that there's over 100-year history of integration breeding with many of the important traits in tomato coming from wild species. And so what I'm showing in this slide is a picture of the red-fruited clade. And so we have Solanum pimpinellifolium, we have Lycopersicum, Solanum lycopersicum variety cerasiforme, so the wild cherry, and there are domestic forms of cherry as well. And then we have an example of a fresh market tomato and a processing tomato. In this panel, I'm showing a picture of some work by Charlie Rick that looked at crossing relationships between tomatoes. And so we have the green fruited clade and then the cultivated or the red fruited clade types over here, and they can all be crossed back and forth with various degrees of success. And then finally I have a pedigree, and this is a small piece of a pedigree that was put together by Jay Scott many years ago. But what I'm trying to illustrate here is two things. One is that the fresh market types are related to the fresh market types, but creating this fruit shape, fruit size, small core, and compact plant involved accessing wild species, these PIs here and here. And interestingly, this occurred twice, once on the East Coast and once on the West Coast, using different pimpinellifolium resources. So even this contemporary type of tomato relied on integressions from Solanum pimpinellifolium. Okay, next slide. So what I'm going to talk to you about is a project that a graduate student in my lab, Deborah Leabouf, worked on as part of her thesis. And I call it the start and end of a variety. So the end is this bacterial spot resistant tomato, and it was bred to combine resistance to 3 different species of Xanthomonas causing bacterial spot. I'll show you the pedigree next, but this is this Ohio 88119 is the susceptible line that makes up much of the pedigree. So we'll go ahead and go to the next slide. So this is the pedigree. So here's the end. This is the multi-resistant line. You can see Ohio 88119 represented heavily, and then you can see some other sources in here. So this Hawaii 7998 is a source of some of the resistance we're working with. This PI 114490 is a source of some of the other resistance that we're working with. And what we did in this project is we sequenced using a resequencing approach anything that has this kind of red circle, and then anything that has the green box we had transcriptome data for. And in most cases, the resistance traces back to either Solanum pimpinellifolium, or in the case of the PI 114490, it's a Solanum lycopersicum variety Cerasiforme type. So let's go ahead and go to the next slide. Yeah. So this workflow is not something that I'm going to go into detail with. What I'm highlighting in the center here is that a key part to any resequencing is alignment to a reference genome. And so we went through this project, through this pipeline, where we're creating this alignment, and then we were looking for copy number variants using a couple of tools. We were doing some SNP discovery, etc. But as we went through this stage of aligning it to a reference genome, we found that we were leaving a lot of our sequence data on the floor, as it were, as much as 20%. So let's go to the next slide. Okay. And so what I wanted to point out is that there is another genome available, and it's a Solanum pimpinellifolium, and this was work done that was done by Zach Lippman's group very early in his academic career at Cold Spring Harbor. And this is considered a reference, although it's really a series of scaffolds. But one of the things that Deborah decided to do was to take her unmapped reads and asked, how many of those could we map to this Solanum pimpinellifolium reference? And then she did some follow-up work to say, okay, where do these actually map in the genome? So she was going from contigs to genome position. So we go to the next slide. Okay, and this is just showing a SNP density relative to Heinz 1706 genome, and I'm only showing 2 chromosomes, and I'm showing everything that we sequenced in this project. And what I'm pointing out is that Deb was able to identify key major genes, so either this RX3 or this RX4 XV3 region that I've boxed here, or an important QTL that also confirmed resistance. And it turns out that this QTL actually was in the resistant line that we were interested in. This QTL was brought into coupling phase with the major gene resistance on chromosome 11. And so there's a finding from the sequence that's interesting, but we could also find at least the main known genes that we needed to. So if we go to the next slide, this is a slide that just gives a graphical representation of the integressions that we found in the multi-resistant line that differentiated it from the line that made up most of the pedigree. And so there are 19 to 25 fragments in this multi-resistant line that are different from the Ohio background. In many cases, we could identify where they were coming from using a phylogenetic approach. And chromosome 11 and chromosome 5 were where we knew that there were some important genes. And then chromosome 3, there's a couple of fragments there that some QTL analysis had hinted at being important. And again, we could trace those in the pedigree. So this 7463 parent has that PI 114490 in its background. Okay, so let's go to the next slide. And all I'm doing here is illustrating how the phylogenetic approach helps us identify the origin of specific regions. And so, here we can see that the region that I'm targeting with this analysis has its origin in pimpinellifolium, and we see that by these RFs in the PI 128216. And then in our pedigrees, they're coming in from this Florida line, and they're coming in from this 7663 parent. Okay, so let's go to the next slide. Okay, what I'm showing in this graph are chromosome positions and then size and total KB of deletions and duplications that were detected in our pipeline that was designed to identify copy number variants. And what I want to point out is where we knew we had some important integressions, so chromosome 5 and chromosome 11, we see a high frequency of these copy number variants, and then chromosome 12 is also showing up here as well. And I'm going to ask you to go to the next slide. Okay, so in the next set of analysis, what Deb did is she took her unmapped reads and she asked, well, how many of those can I map to this set of contigs from the pimpinellifolium? And she found that she had a very high alignment rate. And so 80% of those unmapped reads could be mapped to pimpinellifolium, and 30% of those aligned exactly one time with high fidelity. And then when she looked further, actually looked at gene models within those new alignments, she found several categories of resistant-like genes that were interesting. And in order to do that, she relied on the data set provided from Jonathan Jones's group, and this data set is available as a supplemental file to this BMC publication. And so the point that I'm making here is that the integressions that we're interested in are where we find a very high level of copy number variance, and it's also where a lot of the unmapped reads end up being. And so the regions of the genome that we're most interested in end up on the floor if we're only comparing a single reference. And I'm going to ask you to advance to the next slide. Yeah. So again, the conclusions are that when we use just SNP density comparisons, we can identify three main integressions that are co-located with known resistance genes in QTL. There are about 19 other integressions. 9 of those came from this Hawaii 7998. Some of those other integressions we believe to be regions with QTL that are important for the resistance in this line. Unmapped reads relative to a single reference may contain critical information, including the very genes that we're interested in. Many of those could be mapped to a pimpinellifolium scaffold set. They tended to map to those known integressions, and they also contained candidate genes. So the ability to align sequence reads to multiple references provides a more accurate representation of the genome, even of cultivated tomato varieties, largely because of this long history of integression. And so I think that concludes what I wanted to share with you, and I think Paul is going to do the wrap-up.
Yep. If you can go to the next slide. So yeah, I just wanted to wrap up with talking about a little bit about how NRGene approaches this problem of multiple genomes. As you see, there's lots of sequencing projects that have multiple analyses, partial assemblies, reference assemblies of multiple individuals in a species. Ensuring we can integrate and utilize that sequence information is really important, as you hear, for practical use in breeding and functional analysis. Today, as you see in this slide, we can visualize reference genomes through our browsers, through publicly available browsers, by analyzing, one gene at a time. Yet there's a lot of difficulty in moving between genomes, just in the visualization. In analyzing data, as you've heard, there's also difficulty in integrating these together. Click one more time, please. To move down the path of leveraging all these data sets, NRGene has developed methods to integrate reference genomes, allowing across-genome comparisons to occur. And this is a visualization from an IGV viewer, which I'll show you in a second. Next slide. This integration of information starts with quality assembly, de novo reference genomes. It can be performed on a lot of different workflows. And you heard a little bit from Paul Matthews talking about different assemblies. NRGene performs reference genome assemblies using our two analytic workflows, depending on the complexity and the zygosity of the genomes. The assemblies that come out, in reference to what Paul Matthews was talking about, the assemblies that come out of this are phased. So each scaffold is representative of what's in the chromosome of that organism. The algorithms that you see here, the first one, and if you click again, the next one, is tuned to specific sets of libraries at specific sequencing depths to maximize both the quality for the best value. If you go to the next and the next slide. So once we have multiple de novo assemblies of diverse lines for a given species, or lines that you've heard here may be carrying specific traits that you want to do comparative analyses on, what NRGene has developed is to perform a pairwise analysis of all these genomes to each other. This figure that I'm showing depicts all the possible homologous chromosome segments being compared in a pairwise fashion. The comparison looks across every base of the assembly, and then it determines regions of identity and regions of non-identity in these orthologous regions, and it produces files which allow positioning from genome to genome. Sounds pretty simple, but the complexity of this when you're looking across genomes gets pretty high. Next slide. Then and you can click one more time, by mapping gene models or transcripts to each of the genomes separately, it's then possible to identify these regions, because now we can compare the specific orthologous regions. We can identify copy number variants of each gene model, presence, absence, or even translocations, because we've captured now the entire genome in the assembly, and we can ask, in terms of translocations, where else could this sequence be? So this analysis gets done in pairs against a given, we'll call it a pivot genome or a comparator genome. And okay, next slide. These files can then be utilized by researchers of course, for further analyses. So now we're bringing in the capability, or giving the capability, to position across genomes. NRGene has developed a modified IGV viewer. So this is, IGV is first developed by the Broad Institute, and it's open source. And we modified it in a way to take advantage of these files that I'm talking about. So what we've done is we've given IGV the ability to load multiple genomes into the viewer, and then the ability to automatically position across genomes is become enabled. So those positioning files then become key, and now you can jump from genome to genome and have it positioned correctly. And in addition, what you're looking at here is you can load the pre-computed annotation comparison files to view changes in genes in many different genomes compared to one. What you're looking at the top here is this is all maize, and you're looking at the load of B73 as the pivot genome or the comparator. And we're comparing 4 different maize lines to B73, and the coloring of those gene models that you're looking at. So we're spanning about 4.5 megabases of the maize genome. And so that coloration tells you about the genes in these other lines as compared to the B73 line. So you can see immediately some lines actually lack that gene at that position. Some lack the gene completely. And so when it's not at that position, we can identify it as a translocation and the position across the genome as well. Okay, next. You can zoom in, of course, since this is now based on full genome assembly. You can zoom in and look at sequence information about those genes or the sequences between the genes as well. So this positioning capability and the ability to pre-compute information, the advantage here includes the ability to efficiently, obviously, visualize this, but also investigate multiple reference genomes down to the base pair. The pan-genome structure that we're talking about fits into NRGene's system. And so this goes beyond just doing these comparisons. This system is called Genome Magic, and it allows for the organization and analysis of sequence-based haplotype data. So in the next slide, what we do is use that pan-genome as a positioning or coordinate system, and we capture additional diversity of a germplasm or a given set of lines through not just sequence and matching of those reads to a reference, but we actually sequence and assemble those diverse lines, and then we're positioning into the pan-genome rather than that single reference. So if you heard what Dave just talked about, he used those sequences and went up against a second reference. Here, we're taking large contigs and we're positioning it across all these genomes. So much more efficient in capture of information, much more efficient in capturing haplotype-based sequence information, since everything here is phased. And that was the issue also with Paul Matthews' discussion. We also identify all types of sequence variation, not just the SNPs. And so if you go to the next slide, I'll end here really to help move the effort of developing these pan-genomes. NRGene is working with a number of consortia groups, public and private companies, that are building reference genome assemblies, and NRGene performs the comparative analyses as I just outlined. So the agreement with the consortia is that each member of the consortia pays for the assembly of one genome, and every member receives all the assembled genomes, the mapping file, the comparative analyses, and then if you want, you can download this viewer that NRGene's developed. So to talk more about this and to further discussions, please get in contact with myself or an NRGene rep. You can find us on the website if you're interested in further discussions about this. So I will stop there, and I think we can take a few questions probably if there's still a few minutes left.
Yeah, so Gal again. In the interest of time, we'll make it short. We'll take only 2 questions, but rest assured that everything else will be answered via email, so feel free to send us more questions. So the first question we got is about the number of genomes. Essentially, how do you determine which or how many genomes do you need to sequence in order to capture the complete genomic landscape of a given crop species?
Yeah, I think I can answer that. And I have to say first of all, there is no end to capturing information. Let's put it that way. It's really what is the objective of the project or series of projects? Where do you want this set of information to represent? And so you heard here a little bit about discussions about sets of lines. in terms of tomato sets of lines that were important for breeding. So we can actually measure how much diversity is being captured by bringing in multiple lines and comparing that to other lines that are sequence data from other lines that are available. So there is a measurement way we can do this, but the other piece is, practically speaking, is how much information do you need to move forward in either breeding or functional genomics. And so that's really a further discussion with a group that want to build out this pan-genome.
All right, thank you, Paul. For the last question, I think it's very relevant to Paul Matthews. The question is, can you silver nitrate or ethophone treat hops to reverse its sex and self the plant?
So in hemp, this is commonly used to create what are called feminized lines, so you induce true hermaphrodity. No, so far we're not successful in hops in using silver nitrate or other ethylene inhibitors to induce. We can induce sort of, what can you call it, catastrophic sexual transmutation. You can induce morphologically hermaphrodite, like a female to male floral transition, but they're not, and make some pollen, but it's not competent pollen. So the answer is not yet.
I see.
So we do have, the only inbreds we have are recursive back crosses, state of BC 6 or 7, but so far we don't have